So now we want to look at what happens to the axons of ganglion cells after they leave the eye and form the optic nerve here. Once the axons of the ganglion cell leave the eyes, the main processing stream looks as follows. The retinal ganglion cell axons and support cells combine to form that optic nerve, and it travels down here to the optic chiasm. At the optic chiasm, ganglion cells cross over so that cells with receptive fields in the left half of each retina go to the left hemisphere, while cells with receptive fields in the right half of each retina go to the right hemisphere. So there's this crossover. Our right-hand side crosses over to the right-hand side. I guess that would be the left. And the right here crosses over to the right there, to form these new optic tracts that carry information about one side of the visual field now from both eyes. And they synapse onto, they transfer their information to the lateral geniculate bodies, which is our second major structure in the pathway involved in feature extraction in our primary visual pathway. We'll talk about the lateral geniculate nucleus, and then we'll talk about where cells from the lateral geniculate nucleus transfer their information, i.e. the primary visual cortex, the end of the primary visual pathway. So I thought I'd throw this in. This is a fiber orientation distribution image. It's a picture of the pathways here. And we start off here. This is the optic chiasm, lateral geniculate nucleus, projections onto the visual cortex in each hemisphere. And it was an animation that was generated using diffusion weighted MRI together with a probabilistic algorithm to integrate fiber orientation. And then somebody who clearly did too many hallucinogenics in high school and animated it using colors. So the second stop now is the lateral geniculate nucleus. And it's part of the thalamus, which is a bilateral, one on each side, organ in our brain. And it has a number of visually important structures: the pulvolinear nucleus, the medial dorsal nucleus, and the one that we're focusing on, the lateral geniculate nucleus. And so, as you can see, the lateral geniculate nucleus is a relatively small part of the thalamus. Here's another orientation of it so that you get a different sense of it. So these are the two different thalamic structures. They're connected here with interthalamic adhesion. The medial geniculate bodies here, the lateral geniculate body here, and the pulvolinear areas here on both sides. So when the axons from our ganglions synapse on cells in the lateral geniculate nucleus, we see the following take place, okay? The ganglion cells synapse onto specific layers in the lateral geniculate nucleus. And the lateral geniculate nucleus has center surround configuration cells, and they can be classified as either P cells or M cells or K cells, actually. And the lateral geniculate nucleus also forms a topographic retinal map where the fovea is disproportionately represented. And in fact, this is significant. So when we look at this representation here of an actual slice of a LGN, I believe from a cat, right? Well, one of the things we ought to notice is that there are 6 layers here in the area that's really representative of the fovea. As you move out from the fovea, you start to see the layers shrink in size. Layer 5 here disappears entirely. 4 and 6 become one layer. 3 becomes another layer, so that when you're getting out to the periphery of the retina, there are fewer layers and they are thinner than the layers that we find in the cells that represent the fovea. So focusing on this particular area here, right, we'll notice that those cells fall into 6 distinct layers. Each has approximately the same position in the receptive field. So I illustrated this by here's B in our receptive field, here's B, and here's the line that you could draw down this series of layers where the cells along this line all represent approximately the same position in the visual field. So each layer of the lateral geniculate nucleus receives input from one eye. So layers 2, 3, and 5 receive input from the ipsilateral or same side eye. Layers 1, 4, and 6 receive input from the contralateral or opposite side eye. And then you notice as you move outward, right, you still keep both of these bottom layers, 1 and 2, to some degree, but 3, right, are ipsilateral eye. That's maintained but thinned. 4, 5, and 6, right? 4, 5, and 6, they all condense into one representation as you move away from the cells that represent the retinotopic visual area right by the fovea. So the M pathway goes to layers 1 and 2, the magnocellular pathway, the motion and gross feature pathway. The P pathway gets 3, 4, 5, and 6. And that is a color, fine detail kind of pathway. Now, when they discovered those bistratified cells, what they realized was the bistratified cells actually seem to synapse onto these interlaminar layers here, right? The areas between these individual layers. And so what they realized, they thought those cells didn't play any information processing role, but it looks like the kinocellular layers there, they project onto blobs in V1, in the visual cortex, and they seem to function to provide low acuity visual information and high discrimination color processing in the form of short wavelength on cells.